Hi everyone, this is my second video about the metastable oxygen molecular decay light chamber. If you've seen the first video, you will notice the new chamber is larger than the first one. We went from a 16 square foot platform to 25 square foot platform, with the surface area going from 9 square feet to 16 square feet. Plus we added doors to the chamber as well as one more cell. So instead of 5 cells, now we have 6 cells chamber. We're still in the experimental and development mode of doing things. However, in theory, this chamber can deactivate some virus type completely via process initiated by infrared light generated by metastable oxygen molecule going into its ground state. The type of viruses we believe we can potentially eliminate completely by our process are viruses that do not integrate their DNA into the host ganglion cell DNA. Important examples here are herpes 1, which is the cold sores, herpes 2 is an STD, and herpes zoster, known as shingles. Now, there are two metastable oxygen molecular states. One is called the delta state, and the other is called the sigma state. When the metastable oxygen molecule come down from the sigma and delta state, they emit two different wavelengths of infrared light. These wavelengths of infrared light can penetrate fairly well into the human body when in your nature suit. Oxygen molecule dissolved in the tissue can absorb this infrared wavelength and convert this molecule to metastable oxygen molecules. These metastable oxygen molecules now inside the body are chemically reactive and can damage virus DNA, and with repeated metastable oxygen molecular activation inside the body from those two infrared wavelengths of light, the virus DNA can be made incapable of causing new virus replication due to accumulated damage. Our human cell can have a robust DNA repair enzyme system to repair our DNA damage, but viruses' DNA do not incorporate with our DNA is not so fortunate, and with respect our repeated exposure to metastable oxygen molecular infrared decay light, the accumulated damage to viral DNA can make it unable to replicate and form new viruses. We are so confident of the science behind our chamber that we only ask for donation if our therapy has helped you. And of course, you can get more information by visiting our website www.herpesnomore.org. Thank you.